Genetic modification was once the stuff of science fiction, but in the world today, genetic modification is being used in ever more subtle ways, yet the topic remains controversial. DNA is the fundamental unit of life. It is what genes are made of, and by directly changing it through genetic modification, humans have become more able than ever before to control nature. Genetic modification, or genetic engineering, is a simple process. It involves moving genes from one cell to another. This can be done to replace damaged genes, or it can be done to introduce genes that would never normally be found there. This works because DNA is universal. All life forms discovered use it and understand it in the same way. Take the human gene for insulin. It codes for the protein human insulin, whether it is in a human, a mouse, or a bacterial cell. Once a desirable gene has been identified, it can be cut out using a restriction enzyme. There are lots of different restriction enzymes, and each works at a specific site by recognizing a particular sequence of the bases A, T, G, and C. Another enzyme called ligase is responsible for then sticking the DNA back in its new place. Whilst enzymes can be used to cut and stick DNA, it is still necessary to actually move the DNA between the cells. This is done using what is called a vector. There are two different vectors commonly used. The first is a virus, and the second is what is known as a plasmid. The plasmid is a small circle of DNA that can pass between bacterial cells. By including the desired DNA in either the virus or the plasmid, it can then be moved between cells. Diabetics need insulin to regulate their blood sugar levels. Historically, this insulin was extracted from pigs, but this did not regulate sugar levels as well as human insulin and presented ethical issues. Today, it is possible to produce large amounts of human insulin by transferring the human gene for insulin into bacteria and then growing the bacteria in an industrial fermenter. The human insulin can then be extracted in large quantities and purified. Plants are in a constant battle with diseases, and only by having the right combination of genes can plants resist infection. Genetic modification can be used to add in genes from rare wild types to give an increase in resistance to disease. Whilst this can be achieved through selective breeding, genetic modification is much faster. Many people worry about the use of genetically modified organisms. In particular, they are concerned with the genes spreading into wild plants. Transgenic refers to the transfer of genetic material from one species to another. This is where genetic modification can achieve results that would not normally be attainable through selective breeding. One famous case of transgenics was the transfer of an antifreeze protein from a fish to a tomato, which meant that the tomato would not be damaged by frost, although this particular plant was never commercialized.